Okay, so in the last video we talked about homeotic genes, and now we're going to start talking about the specific homeobox region. Alright, so a homeobox region is going to be a specific 180 nucleotide long sequence that is present in most homeotic genes. It specifies a homeodomain, which if you remember from what proteins are, the domain is going to be the active region that actually does the specific function of that protein. All right, that's going to carry out the function of that protein. Now, they call these collectively, they call them Hox genes, all right, because they're homeobox containing genes. So they call them Hox genes in mammals most of the time. They're present in bicoid, segmentation genes, and they're present in master regulatory genes. And what's great about them is that their sequence has been highly conserved over the course of history of life, over evolution, which means that they probably developed very early in the history of life, and they're very important because they've been conserved over a long period of time. So basically, what is the homeodomain? All right, the homeodomain is going to be specified by the homeobox, but what does it actually do? Well, it's a transcription factor. All right, basically, it's got 3 alpha helices. Then one of those fits into the major groove of the DNA. Remember that the DNA spirals and it has a major big region and then a minor small region. It fits into the major groove of DNA, and in coordination with other proteins, it will bind to enhancers or promoters and actually promote the expression of specific genes. Okay, so it switches them on and off, basically. Now, in the case of transcriptional regulation from within. All right, it's an internal sequence within a cell that starts to regulate the expression of proteins. There is also something called induction, which is basically the regulation of cells from without, all right, from outside the external environment, and this usually means cell-to-cell signaling. Now we've kind of already talked about we've talked about cell signaling before. Induction means that cells signal nearby cells to change some sort of specific way to either express or not express specific genes, and then it brings about differentiation that way. All right, so this means that some cells are going to secrete some sort of signal molecule that is going to be received by the target cell nearby, and it's going to change its pattern, all right, to become something else. Now we call this induction because one cell induces another cell to be something, okay? Now, this is going to be great to look at in C. elegans, all right? So we're going to look at vulva development in C. elegans. Segmentation pattern formation was good to look at in Drosophila, but in the case of C. elegans, it's good to look at cell signaling patterns, okay? So they talk about vulval development in the C. elegans in the book, and it's very, very interesting. It's more interesting than Drosophila. So, anyways, all right. So basically, what they're looking at in the cells that will become vulva, there are six precursor cells, okay, that will form the vulva region of the C. elegans. On top of one of the cells that sits sort of in the middle, you'll see it on page 419, figure 21.17. There is something called an anchor cell, which is going to direct the partitioning of tissue type and pattern formation in the vulva cell. All right, it's called an anchor cell, and what it's going to do is it's going to secrete a inducer to nearby cells, particularly the one that it sits on top of is going to get most of the concentration, and then the ones that are directly next to the one it sits on top of, right? So I'm looking at the second picture here on figure 21.17. Okay, so basically that inducer is going to activate the gene to produce a second inducer, right? So it's going to activate the transcription of a gene to produce a second inducer. And that second inducer is going to bind to receptors on the cells directly next to the main cell that the anchor cell sits on. Okay, so and then that's the ones that are right next to it. Okay, let's see here. the red cell, basically, the first inducer is going to tell it to become the inner vulva. All right. And then as it's activating its expression of genes to become the inner vulva, it's also going to be expressing genes that will produce a second inducer. The second inducer is that green thing right next to it, and that is going to be distributed out to the cells directly next to it in the vicinity, right, which are going to receive it, and then they will start expressing the genes to become the outer part of the vulva, okay? Now, the other 3 cells, the yellow ones, that didn't get any sort of second or first inducer because they were too far away, become epidermis. All right, so what happens if there is, let me give you a question. What happens if there is a mutation in the anchor cell? What if we blast it away? What happens? Yes, you are correct. Okay, so basically what's going to happen is that the anchor cell is not going to be able to give away its first inducer, right? Which means that the middle red cell is never going to get it, and it's never going to be able to give its second inducer. So that means that if it doesn't have any first or second inducer, it becomes epidermis, right? That's the rule. If you don't get first or second inducer, you become epidermis. So if none of them get first and second inducer, they all become epidermis and there is no vulva, right? Which basically means that the worm, cannot expel its eggs into the environment. So they will develop inside the mom, and then they will basically eat their way out. Okay, they'll kill her from the inside out. Okay, so these model organisms are really good to use in terms of, we only talked about 2 of them, but they're really good in terms of looking at concepts. All right, we can generalize broad concepts looking at specific processes through these through these organisms. All right, so first of all, they show us that sequential induction drives development. Okay, inducers depend on transcriptional regulation, and also they depend on concentration. Okay, so we saw that in the case of the Drosophila fly, that the morphogen bicoid is going to be more concentrated on the anterior end than on the posterior. In the case of the vulva development, the anchor cell that gives away inducer to the very closest cell next to it, to the closest cell, is going to have high amounts of primary inducer that will then induce it to create secondary inducer. And whereas the cells that receive neither of those are going to be epidermis, okay? And then this also shows you that inducers work via signal transduction pathway, and that their responses, the responses to the inducer, is usually going to be activation, right? Gene activation of some sort. Okay, so the overarching concept of this is that genetics can be used to look at more than just genes. All right, it can be used to look at development. It can be used to look at evolution, histology, cytology. I mean, it is the controlling factor of all of these big biology questions that we ask. Okay. So the last thing that we need to talk about in terms of animal development is going to be apoptosis. All right, apoptosis is programmed cell death. Basically, it's controlled by the CED genes. The master regulatory gene is going to be CED9. And basically what they do is that they are going to activate the expression of these suicide proteins, which are basically proteins that are going to tell a cell that it needs to die. All right, so the cell is going to start to break down itself from the inside. The nucleus shrinks, the plasma membrane goes away, and then other cells are going to start engulfing the parts so that there's no trace left behind. There's no lysosomes that are just lysozymes that are going to be out in the cell destroying specific things they don't need to be destroying. Okay, so it activates proteases and nucleases. It also activates other cells to start engulfing it. Okay, so you can read about it in the book, but it's good for when you're talking about tumor control, when you're talking about cell death because the cell's just old, or when you're talking about pattern formation in the case of hands and feet. Okay, so we all start with webbed hands and feet, and then programmed cell death tells the skin cells right here on the inside to go away so that we have non-intact fingers. Okay, they're not connected to one another. Now, the last part of this is plant development. I honestly hate plants and their development. It's so boring, but we're going to talk about it anyway. I'm briefly going to go over it because we're going to talk about it more in chapter 35. But basically what it's talking about in plant development is that there are the same sorts of processes to get development. Remember that plants are different from animals because their cells are continually able to become anything in the cell, right? At the apical meristems, remember, which is the roots of the tips. I mean, the tips of the roots of the plant, all right? And the example they give you in the book is called the floral meristem. It's basically 3 layers, okay? L3 is going to be the inside layer, L1 is the outside layer. And basically what it's saying is that L3 is the master regulatory gene, all right? It's going to be the one that controls the organ formation and identification of L1 and L2. All right, now they go over something called organ identity genes, which are analogous to the homeotic genes of the animals. These are going to be the genes that determine what plant structure is going to be happening or is going to be developed in a specific cell. All right, they go over it in your book. I'm done with it, so that's it for now. Okay, I will go over chapter 22 with you guys on Monday. Be sure that you've read chapter 22. Chapter 23 we start on Tuesday. You guys have a test on Thursday. Okay, enjoy the rest of your weekend.